TV and seen videos of baby animals covered in oil and realized it was actually a commercial for Dawn dish soap. So since the early 2000s, Dawn has been known to advertise their soaps as able to clean off oil on not only our dishes, but also on our marine life. Time and time again over the years, we've seen that different detergents can be used to remove oil and make things look as good as new on the outside. But what about on the inside? Oils can be made of polycyclic aromatic hydrocarbons, or PAHs, which are hydrophobic compounds, meaning that they are not attracted to water. So because these PAHs are not attracted to water, they can be attracted to the lipids, such as those found inside our body, meaning that they can permeate our skin. So I want to lead us into a real-world example of how harmful this oil can be. In 2010, the Deepwater Horizon Oil rig exploded and led to the spillage of approximately 134 million gallons of oil into the Gulf of Mexico. This spill was named the largest oil spill ever reported and led to the death of anywhere between 61,000 and 174,000 sea turtles. One of these affected species was Kemp's Ridley sea turtle, which is a critically endangered species only found in the Gulf of Mexico. Now, luckily, there were rescue missions that were conducted that successfully rescued hundreds of sea turtles for rehabilitation. Now, upon initial rescue and after rehabilitation, blood plasma samples were conducted from the external that remain on these turtles to conduct clinical assessments. Now, though these assessments found that there were various physiological derangements in these animals, we don't really know what was happening on a deeper cellular level, which is where my thesis comes into play. So, upon rescue, turtles were visually assessed and were placed into one of 4 oiling categories. This was minimally oiled, lightly oiled, moderately oiled, and heavily oiled. Now, looking at the lightly and heavily oiled categories, my thesis will be focusing on analyzing the protein abundance between these 2 categories. And to do this, plasma samples were thawed and their concentrations were determined. Once these concentrations told me how many proteins were in the sample, I was able to digest them using the enzyme trypsin and turn them into smaller peptides that I could then inject into a mass spectrometer. Now, using this mass spectrometer, I can get identification and abundance data for these proteins that can be used to make protein lists, to which I can then assess whether or not certain proteins are associated with various levels of oil in the turtles. Now, results from this study could be used to, again, deeper our understanding of oil spills and their effects on sea turtles, as well as determine any